So good afternoon, everyone. So today I will be talking about pattern recognition and neuroimaging psychiatry. How do I? Just click, but it didn't. No? Ah, sorry. So this is just the outline of the talk today. So I will start just doing a very brief overview what you've seen in the previous talks about, the supervised learning framework for clinical applications. Then I will discuss some limitations of this framework, and I will present multivariate associative models as an alternative way to look at these clinical problems in psychiatry. And finally, I will present a multiple holdout framework for associative models. I don't know, I'm having problems with just... Huh? Left? Ah, now it works. So, yeah, you've seen similar, I think, slides before and in the morning. And the idea is, with the supervised framework, we have a set of images if we are interested in the classification framework, and then we train our machine learning algorithm to learn, the discriminative pattern between these two groups. And once we have this predicted function, we can apply it to a new image and make predictions. So this is in the classification setup. And this type of approach has been applied to address a number of clinical questions in psychiatry, for example, diagnosis across disease, predicting disease outcome, identifying at-risk subjects, and identifying treatment responders. So if we go to the alternative, similar framework where we treat the problem now as a continuous, we have the regression framework, and we are here, we don't have the classes anymore, but we have the set of images, and then we have the scores that are continuous measures. And again, the idea is that we learn this predictive function, and then once we have a new subject, we can make a continuous prediction now. And this approach has also been applied to address a number of clinical questions in psychiatry, mainly to predict symptoms, intensity for brain scans, and also to predict personality traits from brain scans. So these are, I think, the standard supervised learning framework. I think in psychiatry particularly, there is a big limitation with this framework, which I try to illustrate here in this slide. The fact is that when a psychiatrist or a psychologist decide, on these labels, they do a very big clinical and behavior assessment. And all this information is summarized into a single label, and very often just a binary decision. So there is a lot of information that we leave out of the model that might be very helpful. And in particular, in psychiatry, groups are very heterogeneous, and which means that the labels, the categorical labels, are unreliable. So basically, the clinical and behavior information are not being incorporated in the model, and the labels that we use are very unreliable. And this has been noticed. I think this knowledge that the categorical classifications or the categorical classifications in psychiatry are very limited is well known by the psychiatrists, which has led the NIMH start, a research domain criteria framework to investigate new ways of reclassifying psychiatry. And just to emphasize the problem is that, diagnostic categories in psychiatry that are only based on this clinical and behavioral assessment, because there is no biomarkers so far to psychiatry, they have failed to align with findings from neuroscience, genetics, and they have not been predictive of treatment response. So it's a big problem even within psychiatry is accepted that is a big problem. And the research domain criteria, the RDoC framework, has been trying to develop new ways of classifying mental health disorders based on observable, dimensions of observable behavior and neurobiological measures. And I believe machine learning can play an important role here, and I will try to, over the next slides, to make this point and see how I believe my vision for how machine learning can help redefining psychiatry. So this has been already touched in the previous slide, but I will touch again. One way to approach the problem is from what in machine learning is called multiple source learning, is when we combine multiple sources of information. And of course, in clinical settings, we don't collect often, the research doesn't only collect the structural, functional, but it also collects for clinical populations behavior and clinical assessments. So the idea is to use all this information available. And I see that there are two aims that we can combine this information. One aim, as you've seen in the previous talk, is to build multiple source predictive models. The idea here is just to combine all this different information for prediction, and the final goal here is just to improve diagnosis and prognosis, and I think this is very helpful when you have reliable labels. The other way is to use these different sources of information to investigate, the associations between them, in particular between brain and behavior, and then we hope that by understanding these multivariate associations between these different levels of information might help us to better characterize the mental health disorders and perhaps even to, redefining the labels. So I will show a little bit more information on what I mean by multiple source learning, multivariate associative models. But just before that, I want just to make some analogy or parallel with you've seen in the morning. With the classification, we basically are interested in finding this relationship, either classification or regression model. We are interested in finding this relationship between some images and the labels. And the outcome here, we have a predicted function, which is defined by a weight vector. And once we have this predictive function, we can classify a new example just by projecting the data onto this weight vector. I'm here just leaving the bias outside to make it simple. But okay, and then we, as you've seen before in the previous talks, we find this W in a way that we want to minimize these errors with, when we have a new data set. Now, if we look at from what I'm calling, associative models, by these type of models, I mean models such as partial least square and canonical correlation analysis. Here, we are also interested in finding this association, but now in one side, we still have the brain, but in the other side here, we don't have a single label. We just want to use the whole clinical and behavior assessment, which is like we have multidimensional in one side and multidimensional in the other side. And when we use this type of models, we are interested in finding associative effects. So we find actually a pair of weight vectors, such that when we have such that when we project the data onto this, like the brain data onto the brain weight vector and the behavioral data onto the brain weight vector, we will maximize either the correlation, the covariance between the projections. So yeah, it's just to make some parallel with we've seen before. Now I will go more deeply a bit on these associative models. So associative models here, I mean models such partial least squares and canonical correlation analysis. These type of models, they find a pair of weight vectors such that when we project the data onto these weight vectors, we will either maximize the covariance or the correlation between the projections. So partial least square will maximize the covariance of the projections, and canonical correlation analysis just the correlation of the projections. So these are the optimization problems that these two models are solving. So the PLS, we find U and V that maximize the covariance of the projections, and the canonical correlation analysis, we find U and V that maximize the correlations of the projections. And just to give some notation here, X here, I mean a matrix containing the neuroimaging information, and Y, a matrix containing the behavior and clinical information. So now let's try to see, because I think we're very familiar with the outputs of classifications and regression. So what are the outputs of this type of model? So one output is the multivariate associative effect. So we have, as we have in classification or regression, we have this W. Here we have two weight vectors, U and V, that represents the associative, multivariate associative effect, between the two types of data that we are considering. And once the first pair is found, we can remove this effect from the data by matrix deflation, and we can repeat the procedure to find additional associative effects. And these associative effects can be anchored because the weight vector, each weight vector pair will explain more covariance or correlation in the data than the following ones. So we can anchor these effects. And in the same way that we talked about weight vectors in the classification regression, we can also see here that the weight vectors U and V will have the same dimensionality of the original data types. So if we look at these paired weight vectors, we will identify which features in each view are more related to each of the associative effects. I think these concepts will become clearer once I show the examples, but I just want to give some background before going to the examples. So, and also we can use sparse versions of PLS and CCA to enable us to select the necessary features to describe each associative effect. For example, here we'll have, if we are trying to investigate the associations between brain and behavior, by using sparse approaches, we'll have a subset of brain voxels which are associated with a subset of clinical variables. So just to make a point here, all the issues that you hear today about issues with weight vectors, they will be the same here, in terms of interpretability. But they give us some information, nevertheless, about the model. So an additional information that this type of model give is what I call latent space. Some people also call these models latent variable models, because you can look at this U and V as a latent space. And by projecting each data type onto this latent space, we can see how the relationship between the data varies across the sample. For example, how the brain behavioral relationship varies in a healthy and disease sample. And for example, here, just to give an example, if you would have like the U, the weight vector for the brain, and V, the weight vector for the behavior or clinical variables, once we project the data here, each point here is, it will be, that you have the brain score by projecting the brain data, the clinical score by projecting the clinical data. And the idea is that by looking at these brain behavior relationships, we might be able to find subgroups of patients that have different brain behavior relationships. And the hypothesis behind this idea is that, health people, as a health people, we should have a normal brain behavior relationship. And when something starts going wrong, we might have different brain behavior relationships. You start developing a mental illness. So by looking at this latent space, it can be very informative to find these, or to use these associations to potentially find subgroups of patients and defining new labels in clinical cases. So there has been already applications of this type of approach to neuroimage, so it's nothing new, particularly new. I think what I will be talking here is the type of application and the framework to validate the models, but it has been already previously applied. For example, Leflore and collaborators applied this type of approach to investigate association between genetic data and regions of interest of fMRI data. Avants and collaborators use this type of approach to investigate associations between subscores of the Philadelphia Brief Assessment of Cognition questionnaire and structure MRI data. And Rosa and collaborators use this type of approach to investigate associations between two arterial spin labeling data sets from the same subjects but under different drugs. So this is just to give one. There are actually many more, but just to give some feeling about that there has been some applications of this approach. And I still, what I think is that there are two challenges that I think are not completely solved, that we've been trying to solve when using these approaches. And in different people and different groups, we will address this challenge in different ways. One challenge is how to define the optimal number of variables in each view or in each data type to describe the multivariate associative effect. And the second challenge is how to test the significance of the multivariate effect. And here, the first challenge is basically is an optimization problem, because we will have, if we use a sparse approach, this will boil down to how we optimize the parameters, the regularization parameters, and the second one is how we test this for significance. So I will show an example of the work of my group, actually the work of a PhD student in the group who has tried to address these two challenges. And he proposed here a multiple holdout framework for the sparse partial least square. And the framework he proposed has two advantages. One is that it selects the adequate number of variables to describe each associative effect, and also tests the reliability of these effects by fitting the model to different splits of the data. So he based the approach on a sparse PLS formulation that has been proposed by Witten and collaborators. And it's very similar to the optimization problem I showed before. So we are trying to find U and V that maximize the covariance between the projections. But now we have two regularization constraints here. We have an L2 regularization and an L1 regularization. So we can think of this as an elastic net PLS approach. And we have here two parameters, CU and CV, that they actually control or define the number of variables that we will select in each of the data types. And one way to address this problem, as you've seen already in the morning, is, once we have parameters and we need to optimize, it would be to use nested cross-validation to optimize these parameters, doing a grid search, with nested cross-validation. The problem is that this would be extremely expensive if we want to put particularly a permutation test on the top of that, so it would be basically impossible to do it in a reasonable amount of time. And another issue with that is that often the solution will, as also has been discussed here in the morning today, the type of cross-validation that you choose will have a huge impact on the results. So the tendency normally is, because of the limited sample size that we have, is to use a large number of folds, such as an approach such as leave one out, which has very high variance. Here we use a different approach that will be explained in the next slides, is a multiple holdout framework, which is computationally much more efficient than nested cross-validation. It checks the reliability of the effects to data perturbation, but there is one disadvantage with the approach, is that it really needs a large sample size, otherwise there is a risk of false negative. So just explain here how the holdout framework works. So we start first with the whole data available, and then we leave 10% of the data for holdout, and we have 90% of the data that we call train test set. And then we subsample these from this train test set, 20% for test and 80% for train. So we fit the model to this 80%. So we estimated this U and V, as I explained before, that maximize the covariance of the projections. And then we project the test data and compute the correlation of the test data as a metric. And we repeat this procedure, this subsample, 100 of times, and we do a grid search. So we find the parameter combination CU and CV that gives the maximum average correlations across this subsample here, these repetitions. So once we find the optimal combination of hyperparameters, then we train the model with the whole train test data using the optimal parameters and project the holdout. And then we have the holdout correlation. So once we have that, we can do then the significance test. So we have this original holdout correlation, and now to do the permutation, we permute the data, one of the data sources, and we repeat the procedure, like we fit the model, and then we compute the holdout correlation. What is particularly important here is that we don't need to, because we haven't used the holdout for parameter optimization, we don't need to optimize the parameters during the permutation. So this makes the whole framework computationally I wouldn't say very efficient, but much more efficient if you would have to. So this framework solves the problem of finding the optimal number of variables in each data source, and also the significance test. But I think one question that you might have already is that, okay, 10% seems quite a small number. You know, what if, you know, we have just by chance a very good holdout or a very bad holdout? You know, then it would bias the results a lot. So what we do here is that we repeat this framework 10 times. So every, you know, the whole framework, and then we end up with 10 p-values. And then we need a procedure to combine these p-values. So first we correct them for multiple comparison, and we apply a combined hypothesis, which says that, you know, all the null hypotheses are true. So if any of the p-values are statistically significant, is statistically significant, then we can reject the null hypothesis, or we can reject the omnibus hypothesis or the combined hypothesis. This is a standard way, or has been already applied to neuroimage, this combined or omnibus hypothesis, but has been applied in the context of a region of interest, you know, to combine p-values from different voxels. Has been proposed by Tom Nichols a couple of years ago, and we're just using the same type of approach to combine p-values of this holdout framework here. So now I will move on to an example, because I think it will be much clearer all the theoretical background that I'm giving here when I go to the practical example. This is just the same explanation as before, but just using a different representation. I can come back to this point if someone has questions, but I just wanted to give a practical example of how this framework works and what are the useful outputs that I think for clinical problems. So just as a proof of concept, we apply this approach to investigate the brain-behavior relationship in the ADNI dataset that has been already introduced before. This is a public database with demented subjects with mild cognitive impairment, and old, healthy subjects as well. And the reason why we use this is because we needed a data set that we knew what to expect. So we know these people have dementia. They have memory issues, so we expect to find something in memory-related behavior or clinical assessments. And we also know which brain areas are affected with dementia, you know, like the hippocampus and the amygdala. So you would expect to find also this region. So that was the main reason to use this data set as a proof of concept. So here we use 592 subjects from the ADNI, almost the same number of males and females. We did some standard pre-processing with the structural MRI images, and we used on the brain side the gray matter probability maps. On the behavior side, we use the item levels of the mini-mental state questionnaire, which is a standard questionnaire that is applied for patients with dementia, and it has 5 different domains: orientation, registration, attention and calculation, recall, and language. So what we used here as features were the answers to the individual items of these questionnaires. So these are the results across different splits. So as I mentioned before, we repeat the procedure 10 times. So we end up with 10 p-values. And once we find the first pair of weight vectors, we can remove this effect from the data and then repeat the procedure. So we find a second pair of weight vectors or a second effect. And then we can keep doing that until the point that we don't find significant effects anymore. So for this data set, we found 2 effects. We can see here that the first is quite strong. It was significant even after, you know, all the splits. Even if we correct for multiple comparisons, it's extremely significant, the first effect. The second effect is not as strong as the first, but is also passed the combined hypothesis, and the third effect was not significant anymore. So now let's look at these 2 effects, you know, what type of output this model gives to us. So the first associative effect, as expected, was memory related. So these are the clinical weight vectors. Because it's a sparse approach, we'll select some of the features. Here, as you can see, it selected few domains, orientation, language, and attention and calculation. Oh, and these are the brain weights. And we see here that on the brain side, it selected basically the amygdala, hippocampus. Oh, sorry. I have to, I don't know, maybe the timing is automatic. But what I want to emphasize here is that we haven't said anything to the model that there were these domains. So the model was data-driven. It found these domains, and we know these domains that in the MMSE scores are associated to memory, are related, capturing memory-related tasks here. And obviously, we also know that these areas are related to memory. So it's exactly what we would expect to find. And we also didn't give any information about the three-dimensional information in the brain. So it was really, it basically almost segmented the amygdala and hippocampus. So this was the first associative effect. The second associative effect we believe to be more semantic related. So here are the clinical weights. So it's basically spread over all the domains, and more spread in the brain, but mainly on the left side. And there are descriptions in the literature about semantic dementia that affects more the left side, and we believe we might be capturing this effect here on the second effect. And as I said, another output of this model is the latent space. So we can project the data onto the latent space. One way to look at these projections is across data sources or type of data. Here we have the first, so the memory effect. And then we have the brain scores, the projections of the brain data, and the projection of the clinical or behavioral data. So we can see here that the demented patient had high scores, or high if we look at the absolute value, but lower scores, lower brain scores and lower clinical scores. And then the healthy and the MCIs were here on the top. The second effect, the second associative effect, was basically, we see it was not really able to separate well the subjects. We just see some of the demented subjects here with a slightly, with higher clinical scores. Perhaps either they are a different type of dementia, or they are we believe they are the cases that in later stages of dementia that have a higher score on the second effect. So that is one way to combine. So we look at each effect separately, but combine the data types. Another way to look at the data is combine for each data type, just the brain and the clinical behavior, but here we combine the effects. So here we have the memory related for the brain and the semantic related for the brain. So when we combine this projection, we see is really capturing. So the demented patients, they have like lower brain scores, and in the first brain score, and higher in the second brain scores. So and then it really, this space, it's really capturing the also almost the evolution or the degeneration going from healthy to more demented subjects with the MCIs on the way. And here on the other side, if you look at the clinical data, the first and the second effect, doesn't separate the subjects as well as the behavioral, as the brain scores here. And just, I think that is a little bit of the vision, because here we were not really able to separate the group very well, but we can see sort of this progression of the disorder. And it's possible that by adding additional behavior and clinical information, because here we just use one questionnaire, which was the MMSE. So perhaps by adding other information here, we might find additional dimensions in this space, and then these subgroups might be better well-defined. Or perhaps we'll really find that the dementia is more like a progressive. There are no clusters, and perhaps is what the disease is telling us, that's more like a progression. Slower going from healthy to demented. So just to summarize, I think supervised pattern recognition approaches, have limitations when these approaches are applied to data sets where we have unreliable labels. A particular example is the categorical classification in psychiatry. And I believe alternative models that can integrate multiple sources of information might provide new insights about psychiatric disorders and potentially help us to characterize patient groups. So just to finalize, I want to acknowledge my colleagues and collaborators that contribute to this work. And I have references, we have an open position, but I have the references here if people are interested. And we have also 2 posters tomorrow where we apply this type of approach to other data sets, not dementia data sets, but data sets of adolescents with depression. And if you're interested, I don't have the numbers here, but you can find with the names, and if you're interested in knowing more about that. And I'm happy to answer questions if you have.
Maybe we can take like a question and then we'll move on to the coffee break before they run out of coffee again.
Maybe I take the sugar already.
Hi, very good talks. I'm just curious because the approach that you're using is more toward to a matrix factorization numerical approach, and there's another kind of latter models is more probabilistic modeling approach. So I don't know if you're aware of is there any probabilistic modeling type of application on a similar kind of study?
Yes, very good question. Here I'm talking about the CCA and the PLS, it's non-probabilistic approach, but there is a probabilistic version of CCA. I haven't tried myself, but I believe it might be very interesting for this type of application.
Okay. Thank you.
Hi, thanks for the interesting talk. My question is about the associative. So you had a bunch of weights from behavioral MMC scale and the brain. Could you comment on how those weights varied across different folds, or how do you combine those weights across folds to get those brain maps? Because I assume they will be varying during different folds. Yeah,
we have the different splits because it's not really like folds. We don't, but we have these 10 different splits. How can I say it's actually, we can see that by looking at the p-values. You know, for the case where the p-values were almost all significant, they will be quite similar. And for the case where the p-value is not significant, then we have more variation. But I believe it's with everything, you know, depend on the signal-to-noise ratio, you know, how the effect is. One good thing about this approach is that we can use the parameter space as a diagnosis, because we do this grid search to find the optimal parameter combination, and we look at these plots. Because we have 10 different splits, we have 10 different plots, and they are quite informative already there to know how consistent, I think, for different data perturbations, because each split will be slightly different. But it can vary. Right. But here we are showing the results for the best p-value. This is one single model. Yeah. So I guess the hippocampus voxels will be similar across all the splits, something like that. Sorry? So the hippocampal voxels would be similar across all the splits. Is that? Yeah. The first associative effect was really, really strong and very, very consistent across the splits.